How do we perform multiple sequence alignment? One common way can be if we use common sequence alignment mechanisms to perform multiple sequence alignment. जब हम कॉमन सीक्वेंस अलाइनमेंट के टूल्स की बात करते हैं तो हमें मल्टीपल सीक्वेंस अलाइनमेंट के लिए कुछ ऐसा मैकेनिज्म चाहिए जो मोर देन 2 सीक्वेंसेस को कैप्चर कर सके. फॉर दैट the simplest approach can be if we use hierarchical extension of pairwise alignment. What is the meaning of hierarchical alignment or hierarchical extension? Basically, when we were aligning 2 sequences, input was 2 sequences. Now we have 3 sequences. So for hierarchical extension, it means that we have some parent or child relationship. So what happens? We align 2 sequences which are very close to each other. Then we put them in some common cluster. So cluster means that that is a group which have some common sequences within itself. So one cluster for one group, and then we have more than one clusters, that can be n number of clusters. So if I have 4 sequences, let's say from S1 to S4, so S1, S2, S3 and S4, and if I want to do pairwise comparison between each of them, so I will have 6 total comparisons, such as S1 will be compared against S2, S3 and S4. Similarly, S2 against other 3 and S3 against other 3 and S4 against other 3. So figure wise it would be something like that, as shown in the slide here, that we have 4 sequences S1 to S4, and then when we make them make their comparison with respect to pairwise comparison, it would be something like this, such as that this S1, it is being compared against all 3 others, like this one and this one. So similarly, all other 3 are compared against remaining 3 sequences. So what would be the result of this multiple sequence alignment algorithm? So for multiple sequence alignment, it's not possible that we find out 100% complete or correct result. It would always be an optimal result. What is the meaning of optimal? Optimal is basically the best result that is available. So why it is only optimal result? Why it is not a complete or correct result? Because the procedure for aligning multiple sequences, it's very complex. Why it is complex? Because number of steps required or number of computations required to perform or align these sequences, it's very exponential. Then our limitations of our score mechanism, because at the moment we only have scores for 3 things, which is for match mismatch, for and the other one is for gap penalties. And then our limited understanding of life and evolution. At the moment we don't have our complete idea how life evolves and what is the evolutionary mechanisms which are used for generation of these sequences. So there are different algorithms for multiple sequence alignment. We can say that broadly speaking, there are total 5 algorithms which are used for MSA techniques. So first one is exhaustive search. Exhaustive search is basically an extension of dynamic programming which is used for multiple sequences. Then another is progressive alignment. Progressive alignment has 2 parts. Number 1 part is generation of a guide tree, and the other one is aligning the sequences based on scores from this guide tree. The third one is a conserved blocks based approach. In conserved blocks based approach, we try to find out conserved regions among the sequences, and then we try to align those sequences. It is very similar to approach in BLAST algorithm, which was for finding similarity between word based approaches. Then we have iterative approach. Iterative approach is basically based on genetic algorithms. Again, how it works, it tries to find out similar sequences in the first step, and then it refines its search on each step. And then finally, probabilistic or statistical models such as GIP sampling or hidden Markov models, which are commonly used for this probabilistic approach.